till 2010. Dr. Hao then moved into University of Georgia, USA, as a postdoctoral fellow for 2 years. Also, he moved into CEMET in Mexico as associate scientist for another 2 years, and moved back again to University of Georgia for 1 year as visiting research professional. Now, Dr. Hao is associate research professor and wheat breeder at Institute of Crop Science in Chinese Academy of Agricultural Science, China. Dr. Hao interested about QTL and the genomic mapping analysis for the economical trait in wheat, and he has many prestigious papers in these fields. Dr. Hao's lecture today entitled High Resolution Genome-Wide Association Studies for grain zinc and iron concentrations in wheat and its application in liquid chip development. Dr. Hao has up to 45 minutes for his presentation and 15 minutes to answer your question. For the participants, if you have any question, please raise the hand, or you can write your question in the chat box in your screen. Dr. Hao, you can start your presentation now. You can start, please.
Okay, thank you very much, Said, for your kind introduction. And I also thank Ahmed Salam, Dr. Alam, to invite me to give the opportunity to present in this seminar. So, and today I will give a presentation with the title High Resolution Genome-Wide Study for the green zinc and iron concentrations in wheat and its application in liquid chip development. Let me see. So, and firstly, I'd like to give a very brief introduction of our institute and give a little bit background on the wheat biofortification. And my talk will focus on the genomic studies on green zinc and iron. And finally, we use some genomic result and genotyping by target sequence to develop a liquid chip. So this is the map. You will see our institute located exactly in our capital city, Beijing, in the north part of China. And in this map, you will see the two major rivers in China. One is the Yellow River in the north part of China, and the other is the Yangtze River in the south China. So this is the very our major wheat. I think about 80% of our wheat production 80% of our wheat were planted in this green area. It's about 20 million hectares per year. So our institute was established in 1957. It's already more than 65 years. So, and in 2003, there's another institute of crop germplasm collection and utilization, and one laboratory from the Atom Energy Research Institute. The two institutes and one laboratory merged and reshuffling to a new institute. So this is the current Institute of Crop Sciences, KASP. Our institute has 4 research centers, like the department in university, the crop germplasm and resources, and genomics, and the first one focus on the germplasm collection, conservation, and utilization. And the second research center is genomics and molecular design, focus on the gene cloning and transgenics, and do some breeding by design. And the third one is our department, crop genetics and breeding, major focus on the breeding work. The last one is crop management. So our institute focus on the wheat, maize, soybean, and rice. Actually, Chinese Academy of Agricultural Sciences has one institute in south of China, in Zhejiang province. And they focus only on rice. So we also do some rice work, but not breeding. Only focus on the gene cloning in our institute. So there's some facility. This is the new building actually opened this year. We have an old crop gene bank. The capacity is about you can store 500,000 samples, and with the increasing demand for the collection, so we built a new one, and the capacity is 1.5 million samples can be deposited in this building. And the second facility is the National Key Facility for the Crop Gene Resources and the Genetic Improvement. It's a major building for our institute. Some like gene cloning, gene transgenic, and some work in this building conducted in this building. And we have another building, National Engineering Research Center for the Crop Molecular Breeding. It is exactly this building where I am right now. So, and this building is focused on the crop breeding. So this is a very brief introduction of our institute. I hope there are some students in this seminar, and you are very welcome to pursue your master's degree or PhD degree in our institute in the future. A second, I will give a very a little bit background on the wheat biofortification. So as you know, Dr. Norman Borlaug, the Nobel Peace Prize laureate, he has breeding the Semendorf wheat varieties, and bring make the wheat production and increase significantly, and trigger the later we call the Green Revolution in the world. So that time is the production is the foremost for the breeding. So we call the old production paradigm. Even before the Green Revolution, there's a writer, Rachel Carson. I think she wrote a book, Silent Spring. She think the production is very important, but we have to improve the resource, the utilization efficiency, and protect our environment, because too much the pesticide has been used in agriculture. So there's another paradigm. They proposed the sustainability, and we have with the Green Revolution, the world population has increased almost. I think it's increased more than 100% for the population size. The good thing is the cereal production. You can see in this figure let me see. Yeah, you can see the cereal production increased more than I think it's around 170% has been increased. So it can meet the demand of the increasing population. But for the more nutritious, the pulse, it has increased slowly. Okay, slower than the population increase rate. So that make the world not the hunger anymore, but the scientists found there's many people suffered from the micronutrient malnutrition. That means the hidden hunger. So this is the map. So the worldwide severity of the most common micronutrient deficiencies include vitamin A, the iron deficiency anemia, and the zinc. So they're using the World Health Organization, the data set, the children under 5 prevalence data. So they will see clear the South Asia and the most of the part of Africa are suffer from the macronutrient malnutrition. So a new paradigm is needed. You can see in this table, and I think this is the result about 20 years ago. That time already 2 billion people suffer from anemia. So it is a big issue. So the new paradigm forward agriculture need meeting human demands, need productive, sustainable, and nutritious. So this way can solve the problem is biofortification. So they can meet all the 3 requirements and also cost effective and reachable to the remote, poor rural areas in the developing countries. So this paper, the review paper, was written by the Graham Welch and Roger Welch. So these 2 person actually is the pioneer for the biofortification all over the world. And in 1993, actually that time he's a young scientist, Howdy Boris, is a former program director of Harvest Plus, and economist, working in the IFPRI. So he bring an idea to solve the malnutrition problem using the breeding, and then he went to the 2 scientists, one Cornell University Roosevelt and Robin Graham, and they 3 sit down together and talk about the possibility to breeding the nutrition enriched variety. So this is the initiation of the program for the I think the concept, and they initiated the program for biofortification. In 2004, the Harvest Plus project was formally started. It includes 4 phases. The first phase is from 2003 to 2008. We call the discovery stage. And the second is development stage, and the third is delivery. And from 2018 to now, it focuses on the scale up through partnership, the delivery, the commercialization of the biofortified crops. So I joined Summit in 2012 and left Summit in 2016. I actually participate in this project in that 3 years. Summit has made first cross in 2016 to develop the biofortified wheat. The high zinc source is mainly from some synthetic hexaploid wheat landrace, some spelt, and there's one variety, zinc Sagati, has been released in India in 2014. And this is the first variety released with the zinc enriched. We also do some Qtl mapping and GWAS work. So this is the third part. I did to focus on the genomic studies on green zinc and iron. the first issue we need to resolve, all the program, Harvest Plus program need to resolve is the phenotyping. So the classical phenotyping machine platform is on the left side. It used ICP-OES. It cost it's like low high throughput. It's 40 to 50 samples per day, and cost about $15 to $20 per sample. So also it is seed destructive. You cannot use the seed anymore after testing. So the Harvest Plus program, they set up the new facility we call the XRF machine. It's seed non-destructive. It is a very high throughput and also very low cost. So one person can run like 200 to 300 samples per day. So it has been promoted to use in the Harvest Plus program globally. So it's very easily used, and I think there's no training, very basic training, and the result is reliable. They tested the correlation between the classical method and the new method. For zinc, it's very the correlation is very high, and for the iron, it's also acceptable. So it's now used as a routine protocol for the micronutrient measurement. The second one is the soil heterogeneity. So if the soil is not uniform for the zinc, then the genetic studies cannot be done. All the data is not appropriate and cannot be used. So CIMMYT has applied the common zinc fertilizer and year by year and to homogeneous of the soil to do the genetic studies. I think it's very important for the research on this micronutrient element. The real genetic difference become highly visible with soil enrichment. You can see the fertilizer is not used. The difference is very minor. It's 5 ppm. When the fertilizer will continue applied, the difference become highly visible. So we do some QTL mapping work. This is the first study for the green zinc or iron concentrations in CIMMYT. We work on real population, PBW343 and Kenya Savada. We found two major QTL. The 2B QTL actually has the pleiotropic effect, not only increase green zinc, but also increase the southern kernel weight. We have some other population from the spelt wheat, from the synthetic hexaploid wheat. We find some locus. So, and we continue working on several population, mapping population to detect the significant loci for the green zinc. At that time, we only have marker. We don't have many information on the gene sequence. But until the 2018, the Chinese Spring reference genome sequence were released. Then we can have the access to many genes and annotations from the genome sequence. So we can, like, we know there's many genes in the model plants, like rice and Arabidopsis. They have many genes related to the zinc or iron homeostasis. So we transform this gene, we blast this gene in the wheat genome sequence. We find the orthologous genes, and we found about 254 genes. They have the potential role for the wheat, for the zinc homeostasis in wheat. And then we make a collection, do the review work. All the about 140 loci has been reported in weight for the zinc. There's another 11 known genes. So we have the opportunity to integrate all the QTL, all the orthologous genes, and the cloned gene in one picture, in one map. So this is use the 2A, 2B, and 2B as example. You can see there's some gene or QTL being reported in different studies. So it means it's pretty stable and useful. So this is the whole picture. We have made all the 21 chromosomes with the gene or the candidate gene or QTL. So with this picture, we can know which chromosome is important for the with biofortification, and which gene it probably can be useful in the breeding. So we have the 452 weight lines. We just select some of them, 166, and genotyped by 2 genotyping chip, 660K and 90K. It's different solid chip. So we have the high density molecular markers. Then we use the XRF to determine the concentration of zinc and iron. We run the GWAS, and in this case we can find some gene, candidate gene or loci. Then we design a liquid chip, the gene-specific liquid chip. We can run the rest of lines to do the candidate gene association analysis and do the genomic selection. And finally, we find some new marker, new gene, and a new germplasm that can be used for the breeding. So I give brief, introduce the heritability for the green zinc and the green iron concentration. It's okay, it's high, and 0.71, 0.72, it's acceptable because it's for this kind of treat. It's complicated. Many the environmental factors affect this treat. The key is the application of the zinc fertilizer. It's very important. We run the routine analysis to run the genome, the GWAS, and out of the 750K markers, we have about 50% of the marker are polymorphic and can be used for the analysis. We use 3 models to run the GWAS, and finally we found 25 significant loci for the green zinc concentration and 16 for the green iron concentration. So you can see there's some mark trait association very close to some known gene. So we can find the 28 candidate gene were related to the zinc or iron uptake, transport, storage, and regulation. We give a very brief model for the accumulation of the mineral from the root to the head to the seed. And for the expression profile of the candidate gene, like uptake, the gene related to uptake, it is high expression in the root tissue. So it means it's makes sense for the gene's function. And importantly, out of the 28 genes, we see 11 genes. 11 genes actually are putative with orthologous of the rise or Arabidopsis gene. It already cloned, knowing function to the zinc homeostasis. So that means the GWAS data, the GWAS has a significant, very strong power to detect the loci. So like this one on the 3A chromosome, this is the weight multi-drug and toxic compound extrusion transporter gene. It's one of the candidate gene. So it is ortholog of rice gene OSPZ2, and it is related to the iron transportation. So we validate this gene using the biparental population. We have ZM175 and LX987 real population. In this region, actually there were 9 genes. Only the gene OSPZ2 has polymorphic between the 2 parents. And also the marker data showed this gene significantly associated with the trait in 4 environments. So we can see this 3A gene must be the candidate. So we are working for the functional validation right now. There's another 2D QTL. It's also validated into another population, significant associated with the trait. But the 2DL, we found that it is located in suppressed recombination region. It's a little bit difficult to do the fine mapping. We predicted 1 gene because this locus increased green zinc, but also increased southern clonal rate. So, but it is important for the breeding because there's a low recombination. They can transfer together and increase green weight and green zinc together. The last one, probably not last one, we have another locus, the 6DL QTL. We also use 2 populations to validate another new population, the GD8 and the AK58. We found 2 genes in this region, but one has no polymorphic, the other has a very abundant variation. It's the nicotin anime gene, called NAS gene. It was predicted to be the candidate. So now we are do the transgenic and gene editing plans to validate the effect of these genes. We hope next time we can report this result and find the candidate gene. So the last part I will talk about the liquid chip development using the genotyping by target sequencing. So the loci selected for developing liquid chip. Eventually, 77 gene-specific SNP corresponding to 36 genes, including 6 known genes as the reference, and was selected to develop the platform. All these genes are from most from the GWAS we have done previously, and some from the gene cluster, major cluster for the green zinc. So this is the 5 reference gene and 1 functional gene, and some other are gene-specific locus. Actually, there's some are no polymorphic, and we finally select 77 genes, SNPs, and test in with known genotype. And the chip was developed using multiplexing PCR technology. That means we put all the markers, we design primer, and we put all the primers together in 1 tube and run the PCR. And finally, we use the next generation sequencing. We have the sequencing and do the bioinformatics to have all the sequence variation for the SNPs. it takes 5 months from the establishment to the completion of the genotyping target samples, and the cost is pretty low. This is some data. Finally, we have the chromosome distribution of the markers. Actually, there's some chromosome has more markers, has more genes, not evenly distributed, and the missing rate and the heterologous rate are acceptable, right? It's pretty low. So this liquid chip high throughput. As I mentioned, it can be genotyped by one amplification assay, and low cost is 0.05 dollar per sample, and high accuracy because we have the reference gene. The genotype data is completely consistent. So we phenotype the large population for all the germplasm for more than 400 in 6 environments. We have the phenotype data, and then we run the liquid chip. So we do the candidate gene association study. Finally, we can find 19 genes significantly affected the green zinc concentration, and 15 genes affect the iron. So most of the gene actually related to, because it was developed from a smaller population. That 1660 population actually is in this large panel. There's some genes is very stable detected. Some of them is the same as the GWAS, is the same gene detected in the previous GWAS study. So we can see there's significant additive effect. We can cumulative. The more favorable allele the cultivar has, and the higher the green zinc concentration the cultivar will be. So I think we have like about 20 genes, all we call the 20 locus. It is important and also enough for breeding. For the breeding, if we can pyramid those locus, then we can have the higher green zinc. So this is some twin varieties. You can see because most of the variety are from China, and there's some high zinc variety. They have some novel locus we cannot detect. So this is some work we can do in the future. But for Chinese variety, we know the profile for which QTL they have. Then we can pick the parent purposely and pyramid those locus. The same thing for the iron. We have some variety actually. It's both iron and zinc is high iron and high zinc. So it can be used for improve the two traits together. And the genomic selection based on the liquid chip, we test actually we use the 402 cultivar as a training population and 50 advanced line as a breeding population. We run different models. For the green zinc, different model, we see there's some model, the prediction can be as high as 0.51, and for the green iron, it can reach up to 0.73. So we think this liquid chip can be used immediately in breeding for the gene stacking or the genomic selection, because this is a complicated trait. It has many genes affect the trait, and we can pick some major one like 20 genes, and it's the cost is low. As I mentioned, it's 0.05 dollar. It's 5 cents per sample. You can run in a pretty large population to have the individual plants to select and do they have the high chance to have more favorable locus. So that's all I'll present today. Finally, acknowledgement to from 2012 to 2016. I thanks the Summit Global Wheat Program. They provide a lot of the population and the breeding work. We work together, and the funding is Harvest Plus and the Gates Foundation and CRP Wheat, also the National Agricultural Research System. From 2016, I joined KASP, the wheat breeding program, and particularly I would like to thank my two graduate students, Jing Yang Tong and Mengjing Sun. They conduct a lot of work in I presented today, and also I thanks collaborators the Awis Rashid in Pakistan, and the collaborator in the different university, Zhang Yelun and Liu Lianzhen, and some other colleagues from different academy university. And finally, the funding was National Key Research and Development Program China. So that's all my presentation. Thank you for your time, and I will be happy to answer any question if you have. Thank you. Hello.
Dr. Said?
Hello. Yeah, hello. Yeah, thank you very much, Dr. Ho, for your nice presentation. It's really nice presentation. And for audience, this is the time for a question. You have 15 minutes for a question. If anyone has a question, he may please can raise the hand, or he can write his question in chat box in his screen. I think we have here one question. So maybe someone, his name is Sovereign Goal. His question, he asking about the wheat species you have used, Dr. Ho. Is this Triticum acetivum or Triticum durum?
I beg your pardon. Can you repeat the question? You mean the tetraploid wheat?
Yeah, he asking about the wheat species. Is the Triticum acetivum or Triticum durum?
It's dicoccum. Yeah, it's I think it's wild tetraploid wheat. It's not durum wheat. Usually some wild species rather than the cultivar species, more green zinc and iron. The same actually is growing many germplasm. They have a very large collection of germplasm worldwide, and they initially found some like the sprout wheat, the tetraploid wild amber, or some synthetic hexaploid wheat are rich for the green zinc and iron. So then they also there's some landrace. Then they start to use the integration, the make cross and do the selection, and now they can use the markers. And probably 10 years ago we don't have marker. We just do do the cross and the generation advance to get the advanced line. When the line is stable, we measure. We use the high throughput phenotyping tools to measure the concentration of green zinc. Then we can do the yield test or something. I hope I can answer your question.
Thank you, Dr. Hao. I think Dr. Sovetgel, he got his answer. Yeah, I think Sovetgel has another question. He asked, would it be possible to conduct the same research on hexaploid wheat? And also he asked, is it possible to use GWAS with a liquid chip or biparental POP GBS?
Okay. Probably I misunderstand the question the previous. All the study we conducted, except the one we conduct in Semito. In Semito we have like we have one tetraploid mapping population, and all the population for the GWAS study is hexaploid wheat, AABBD, hexaploid wheat, and you already see in the presentation we have like the one gene has 3 copies, like 2A, 2B, 2D. We have found 3 genes, and we design the genome specific primers. So yes, it is possible. We actually done is focus on the hexaploid wheat. We run the GWAS with the liquid chip also, because it is very cheaper. We have a big population, more than 400. But if we run the like high density the solid chip, it costs too much, because one sample for the 660 K chip, the chip, one sample is about I think more than $200, more than $200 in China. So we don't have that budget to run the whole population. So we pick 166. But for the rest of the cultivar, we run the liquid chip. It's much cheaper. The liquid chip, it's very cheap. For one sample, like we have 70. I think it's about 70. It's less than $0.5, half dollar. Yeah, for one sample.
Thank you, Dr. Ho. I think also Dr. Sovetgel, he wants to ask you if it is available for cooperation or collaboration with you. And I think maybe you can tell him about if there is any available collaboration or not.
Sure, yeah. It's open. It's open to everyone. We can collaborate. You can use our liquid chip, or you can. We have different projects. We can work together. You can email me, or you can contact Dr. Ahmed Salam. We have very close connection with each other. Yeah, we can working together, either on the biofortification or some other project.
Also, I think Dr. Sovetgel also he wants to ask if you could suggest a specific company for liquid chip.
For this biofortification chip, actually we work together with a local company. We design the primer, and they have the facility to run the next generation sequencing. So I think it's very easy to find a partner to run the the liquid chip. We have like we have a package, each primer package. It can be used for several years. So if you feel interested on this project, you can, if you have some like a genotype and you have the phenotype data for the zinc, green zinc, and you can send us DNA, we can run the genotype data for you. I think it's very low cost. We can work together. If you have the phenotype data for the mapping population or the natural population.
Yeah, also he asked this will be including bioinformatics.
Yeah, yeah. They have the software to analysis. It's a routine. It become very simple right now. We have some software to, because we have make different genes, specific markers together. They have the label. They have the label. They can give the signal which SNP, which variation is which gene. So they have the bioinformatic pipeline.
Thank you very much, Dr. Ho. I think also Dr. Sovetgel, he need to download this presentation. I think this video or the link for this video will be available on YouTube channel after we get approved from Dr. Ho for that. And for Dr. Sovetgel, if you need any further question or any contact number or for anyone from the lecture, please you can email us and we can send you the enough information you need. Yeah, anyone has any question? I think there is no question more or no more question. Dr.
No, I
Ahmed
have
Salam.
question, Dr. Said.
Yeah, Dr. Ahmed Salam, I remember you. You can ask.
Yeah. Okay. First of all, thank you, Dr. Hao, for this really very interesting lecture and the very useful information that was given to us. So I have two questions. For GWAS study for zinc and iron, have you found a common marker that catching both elements?
Yes, yes, yes.
Okay.
The green zinc and iron usually highly correlated with each other. There are some common, but there are some like specific.
And this like SNPs like major effects like R square major effects or minor effects on the both elements, the genes that you detect, it's like has major effects. Did you measure R square for each marker?
Yeah, we measured the R square for the GWAS, and also we typically validate those MTA in a biparental population. We usually validate in a biparental population. In that case, yeah, in that case, it will be show the real effect.
And regarding to liquid chip methods, it just used 77 genes. Are there limitations for using number of genes? Can we use like 5 genes or 100 genes?
Yeah, it's flexible. Yeah,
it's flexible.
It's a primal. It's a package. You can add or you can remove. It depends. Yeah.
And how is possible that this liquid chip methods can work for other crops like barley, maize, or it is designed for wheat?
No. You can use any crops but different primers. You use different package.
Okay, because it's
Yeah.
really very really cheap. I mean, Yeah, the KASP, I mean, the best way for validating now SNP marker is to use KASP marker, and the KASP marker is really one marker can cost much money in terms of
Yeah.
euro or dollars.
Yeah.
Because I use the KASP for some of my projects, and we paid a lot of money
Mhm.
to genotype few genotypes, like. So this is very quite interesting and will help for making validations
Yeah.
for markers and genes detected by GWAS. Extremely
Mhm.
interesting. Yeah. Thank you so much. I thank you for answering the questions.
It's my pleasure.
Yeah, I think there is no further question. Maybe someone has another question. Oh, Dr. Ahmed Salam. Yeah, I think there is no more question. Thank you very much, Dr. Ho, for your nice presentation. And thanks for all audience for their participants. And I will let the speak to Dr. Ahmed Salam if he want to announce something about the certificates or any other information regarding to MGBS.
Okay, thank you, Dr. Said. For the email of Dr. Hao, we included his email address in the poster. And please, Dr. Hao, could you please write your email in the chat, because some people asking for providing your email. And regarding to the lectures, we will ask Dr. Hao if he allow us to make the video, the lecture public, and we have a channel of MGBS. Due to workload, maybe it will be uploaded later. So just you can subscribe to the channel. So once we uploaded the video, it will I think the website will send you a notification that the available the video is available for you. And for the certificate, we will send an email because we will have another lecture next week for Dr. Hiba. And please follow up your email. We will send an instruction how you will get a certificate for attending the MGBS seminars. Okay, I finished, Dr. Said. Thank you. Dr. Said? Oh, I think we may lose the connection.
Okay, yeah,
So,
okay, yeah.
oh,
Yeah, okay, Dr. Ahmed. Yeah, thank you for all. And also at the end of this seminar, I want to express my deep thanks to Dr. Ahmed Salam and the committee of organization for this MGBS for this great efforts to arrange and organize this meeting. And we hope to meet you again for the next seminar and the next years for MGBS. Yeah, thank you for all.
thank you, Dr. House. Thank you so much.
Thank you.